It's a pleasure to be here. I would like to thank the organizers for the invitation. It's actually my first time in Brazil, so I'm super excited to be here, except I just got off the airplane a few hours ago, so I'm a little bit tired, but I'll do my best to make this entertaining and informative. So what I'm going to do, I'm actually giving 2 talks today. And so in this first talk, I'm going to give you an introduction to what we call geroscience, or the biology of aging, which is something that I've been studying for the past 20 plus years. Most of this talk will be geared towards human biology, but I know that this is a veterinary expo, and I just want to make sure everybody recognizes that everything I talk about relevant for human biology is equally relevant for companion animals and veterinary biology. And I'll try to show you some specific examples. And then in my second talk, which will be at 1 p.m., I'm going to talk specifically about something called the Dog Aging Project, which is the largest longitudinal study of aging in the world that includes more than 50,000 pet dogs in the United States. So that talk will be focused very much on companion animal health and longevity, and dogs in particular. Okay. So I don't know if anybody here knows who Peter Attia is. I know a few people do. If you don't know who Peter Attia is, I'd encourage you to check him out. He's a pretty famous physician and podcaster in the United States in the longevity space. I have done several podcasts with Peter, so this is a picture from one of the more recent podcasts I did with him at his house. And the reason I'm showing Peter here is he has sort of coined the phrase medicine 3.0. And I like that phrase. And I'm going to talk today about medicine 4.0, which is kind of the next iteration of medicine 3.0. So Peter does a really good job of kind of explaining the history of the practice of medicine, right? So medicine 1.0 was like the Dark Ages, leeches, and not very scientific. Medicine 2.0 really was the birth of scientific approaches to medicine. And that's really what we've been practicing for the last 150 years. It is very much focused around individual diseases in isolation, typically waiting until people get sick to really try to do anything to take care of them and try to treat the symptoms, optimally cure the diseases. But really what we end up doing is usually treating the symptoms of the diseases. So that's medicine 2.0. That's what we are. That's where we are today. We're starting to transition to what Peter calls medicine 3.0, which is a more proactive, preventative approach to keeping people and hopefully companion animals healthy, rather than waiting until they get sick to try to do anything about it. So medicine 4.0, though, is what I'm going to tell you about today. And I sort of am of the opinion that maybe we should just skip medicine 3.0 and go right to medicine 4.0. And hopefully I can convince you why I feel that way. But I think there is definitely a desperate need for more proactive, preventative approaches that are designed to keep people and our pets healthy instead of treating disease. So I want to talk about a few key concepts that will be important during this talk today, and give a few definitions. So lifespan, I think, is pretty easy. Everybody kind of gets this. Lifespan is the length of time from birth until death. Health span is a trickier concept. And so lifespan is quantitative, right? We know how to measure the length of time from birth till death. Health span, I would say, is semi-quantitative or qualitative, but it's an important concept. So health span is the period of life spent in good health, high functioning, hopefully free from chronic disease and disability. The reason why I say that health span is qualitative rather than quantitative is we don't have a great way to measure health span. We don't have a consensus definition for when health span ends or when health ends. In fact, I would say health is not binary, right? We don't wake up one day and go from being perfectly healthy to being completely unhealthy, right? It's a gradual, continuous sort of variable. And so I tend to shy away from trying to talk about measuring health span, but I do think it's a really, really important concept. The third definition I want to give you is kind of central to my talk today, and that is geroscience. And geroscience is the science, the field of research that connects the biological mechanisms of aging, and I'll tell you what I mean by that in a minute, with all of the functional declines and diseases that go along with aging, okay? So geroscience is really that intersection between biological aging and the negative consequences that go along with biological aging. Okay, two other terms that I want to mention before I dive into this are the difference between chronological age and biological age. So again, chronological age is really easy, right? Number of candles on your birthday cake, the length of time that you have been alive. Again, we all know how to measure chronological age. Biological age is a little bit of a different concept. Biological age can be different from chronological age, and it's really the age of your tissues, your cells, your organs in terms of their physiological function. And I think one way to really appreciate this idea of biological age is to just think about the way that dogs age relative to the way people age. Everybody here, I'm sure, is familiar with the idea that 1 human year is about 7 dog years. All that really means is dogs age biologically about 7 times faster than people do. That's not a perfect equivalency, and I'll come back to this. But that clearly, I think, illustrates the idea that from a biological perspective, dogs are aging faster than humans are, but chronological age is the same, right? A chronological 3-year-old dog has been alive the same amount of time as a 3-year-old person. Biologically, they are very different ages. Okay, so hopefully everybody gets that. And again, for those of you who are veterinarians, you will appreciate this. And probably most of you, if you've ever had dogs, will also appreciate this. Even within dogs, there's this very interesting relationship between body size and rate of biological aging, right? Big dogs age biologically faster than small dogs do. They have shorter life expectancies, and they get most age-related diseases at an earlier age than small dogs do. So this idea of biological aging can vary across species, right? The difference between humans and dogs, but even within a species, you can have pretty dramatic differences in the rate of biological aging. And I would suggest that we all know people in our own lives who are aging biologically faster or slower than average, right? And that's affected by a lot of things. It's affected by genetics, but it's even more strongly affected by environment, lifestyle, smoking, pollution. All of those things influence the rate of biological aging. Okay. Oh, by the way, this is my dog. His name is Dobby. He's named after the elf from Harry Potter, for those of you who are familiar with the Harry Potter series. Okay, so Medicine 2.0, I already sort of defined, right? Medicine 2.0 focuses on individual diseases in isolation. And I do think it's useful to mention that this is really the culture of medicine, but it's also the culture of basic biomedical research, and it's the culture of pharmaceutical drug development, right? From the very, very beginning all the way through to clinical practice, we focus on individual diseases in isolation, and we typically try to develop therapies that will treat individual diseases in isolation. And these are some of the major causes of death and disability in humans. So I want to first talk about why we need to change. And then I'll talk about how we change. So I don't think Medicine 2.0 is going to be sustainable for very much longer. In almost every developed country, life expectancy is starting to plateau. It has gone up pretty dramatically for the last several decades in most developed countries around the world. But we're starting to see this plateau. And in many developed countries, including the United States, health span is actually decreasing. There are more and more people suffering for a longer period of time with one or more chronic diseases. The United States Center for Disease Control estimates that 60% of Americans have at least one chronic disease. The number I could find in Brazil is 45% of Brazilians have at least one chronic disease, okay? Those numbers are going up, and that's a problem. It's especially a problem when a large fraction of healthcare expenditures goes to treating people with chronic disease. Because if the number of people with chronic disease keeps going up, that fraction of healthcare expenditures that goes to treat chronic disease is also going to keep going up. And again, I don't know what the number is in Brazil. In the United States, estimates are it's 90% of healthcare expenditures. And I don't know when the system breaks, but I can guarantee you that number can't get to 100%. So we need to do a better job of keeping people healthy rather than keeping people sick, keeping people who are sick alive. Don't get me wrong. We should treat disease, and we should keep sick people alive. If you're sick, we should treat you. But wouldn't it be better if we kept you from getting sick in the first place? And that's really, I think, the promise of Medicine 3.0. So this is what's happening right now. Medicine 3.0 has the goal of what we call squaring the curve, pushing out health span so that health span gets closer to lifespan, right? Again, the idea here is not so much about making people live longer, although that's a byproduct. It's really about pushing the diseases of aging back as far as we possibly can into old age. And I think that the key tenets of Medicine 3.0 really are, we're still going to treat sick people, so we're still going to do Medicine 2.0, but there's going to be more of an emphasis on proactive screening, identifying pre-disease early, personalized biomarkers, so we can not only identify what your trajectory is, but develop therapies that are personalized to you based on those biomarkers. Obviously, lifestyle is hugely important. That is probably the most important factor for maintaining health for most people. And then, when appropriate, medications and supplements, but in a preventative way. This is the fundamental difference, again, between Medicine 2.0 and Medicine 3.0. Rather than waiting until people are sick to treat their disease, we can use some of these same medications to keep people healthy. And I'm going to talk more about a specific case example in a few minutes. So this is Peter's book where he really goes into this concept of Medicine 3.0. Again, I love this book. I think it's a great book, and I highly recommend it if you're interested in this topic. Okay, but I do want to say Medicine 3.0 is insufficient to achieve this goal, and I'm going to tell you why. I think it can get us part of the way there, but it's not going to get us to where we need to be, where I think we can get to. And that's where Medicine 4.0 comes in. Okay. So again, we think about diseases in isolation. And one of the ways we can illustrate why we need Medicine 4.0 is to think about what are the risk factors for these diseases, okay? And in particular, what's the greatest risk factor for each of these diseases? And it turns out all of them have the same greatest risk factor. And it's not how much you drink, whether you smoke, how much you eat, it's how old you are. So it turns out that age is the single greatest risk factor for every single one of these major causes of death and disability. And so it follows from that the biological mechanisms of aging that determine biological age are in fact the greatest risk factors for all of these things, and I would argue causal for the increase in risk that we see with age for these diseases. And so if we can understand what this is from a mechanistic perspective, that gives us the opportunity to actually develop therapies that can modulate the biology of aging in a way that will slow the onset and progression of all of these functional declines and diseases simultaneously, at least conceptually, okay? I'm not going to suggest to you today that we know with 100% certainty how to do that in people, although I think we're close. But conceptually, if we can understand the biology of aging sufficiently, we can accomplish that goal. Okay. And if we can do that, again, we should slow the onset and progression of all of these things at the same time. So I told you that age is the greatest risk factor for all of these diseases. I think probably most of you believe me, but if you don't, it's useful to look at the data. So again, this is data from the US Center for Disease Control. So this is data in the United States. It's probably not too different in Brazil. Looking at what most physicians will talk about in terms of risk factor for chronic diseases. The top 3 killers are heart disease, cancer, and COVID-19, and this is from 2021. Alzheimer's is down the list. It's like 6th on the top 10 killer list in the US. So these are 4 of the top 10. And the risk factors that you will typically hear about for cancer, heart disease, infectious disease, and Alzheimer's are, do you drink? Do you smoke? Are you obese? It turns out for Alzheimer's, there's a genetic predisposition, the ApoE gene, that increases your risk about fourfold. So you can all see these risk factors increase risk somewhere between 100%, one-fold doubling of risk, to 400%, fourfold. That's the sort of the magnitude of risk we're talking about, okay? So now if we compare this to the increase in risk from 40 years of aging, that's what it looks like. So I told you age is the greatest risk factor, but I think this illustrates just how big the risk is, right? So again, I'm not saying those other risk factors aren't important, but if we can tackle this, the impact we're going to have is an order of magnitude greater than what we can do by tackling those other risk factors. So again, I think the promise is there if we can actually solve this problem to have pretty large effects. Okay. So you can also look at this from the perspective of life expectancy. So again, the US CDC is very good of tracking when people die and what they die from. And so several years ago, a demographer named Jay Olshansky carried out what is a pretty simple thought experiment. And he just asked the question, if we look at the CDC data and we take out all the deaths due to heart disease or all the deaths due to cancer, what does that do to the life expectancy of a typical 50-year-old woman in the United States? And this is what we would expect if I had a pill today, that I could give you that would cure all forms of cancer. This is what you'd expect, about a 3-year increase in life expectancy. Now, I don't know if you think that's a big number or a small number or about right. I know when I first saw this, I was like, I can't believe the impact of curing all forms of cancer is only 3 years. But it turns out it kind of makes sense, because if you cure cancer and don't do anything about heart disease and infectious disease and dementia and kidney disease, all of those other things continue to increase exponentially at the same time. You get about the same effect, a little bit bigger from curing heart disease. Even if you cure both, it's about 7 years of life expectancy. So then we can ask the question, what would the impact be if we can modulate the biology of aging? And again, we haven't done this in people yet, okay? So this is now comparing to what is routine in laboratory animals. And I'm actually going to show you some data. So you take my word for it right now. This is easy to do. We know how to do it in rodents in the laboratory. Modulate the biology of aging, increase lifespan by anywhere between 20% and 50%, okay? If you did that in people, you would get a corresponding 3-decade increase in life expectancy for that 50-year-old woman. So again, hasn't been done yet, but the potential is there for much larger impacts on life expectancy, with the added benefit that most of those years are expected to be spent in good health, because we haven't fixed just one thing. We've fixed a bunch of things that go wrong with aging because we've targeted the biology of aging. So that's the promise. And again, I want to make the point, if we can do it in people, we can do it in companion animals. If we can do it in laboratory animals, it's very likely we can do it in companion animals today. Okay. So the other thing I want to tell you, I know I'm up here talking about slowing aging, right? And that might sound like a little bit like science fiction to you. So I think it's important to appreciate the science in this field has come a significant way to understanding the biology of aging in the last 20 years. That's about how long I've been in the field. I started working on aging actually 25 years ago now. So I certainly don't want to suggest we know everything. There's a lot we don't know, but we know a lot more than we did 20 years ago, to the point where we can actually point to mechanisms. So this is something called the hallmarks of aging. This is a collection of 12 molecular biological processes that seem to be shared about the aging process across the animal kingdom, okay? So you can go from very simple organisms like nematode worms and fruit flies, to mice, to dogs, to non-human primates, to people. These hallmarks of aging seem to contribute to the biological aging process across that entire spectrum. And so they include things like genomic instability, which would be DNA damage, shortening of telomeres, epigenetic changes. Some of you may have heard about the epigenome in the context of aging. for today's talk, it doesn't really matter exactly what these things are. It's sufficient to understand that we know enough that we can actually start to target one or more of these processes and have impacts in laboratory animals on biological aging. I do want to mention these hallmarks are interconnected. That's what this sort of, the arrows in the middle are meant to represent. And there's a biological network of interacting proteins and metabolites that underlies the hallmarks of aging. And we know what some of those factors are, and we know what some of the key nodes in this network are. And I'm going to talk a little bit about one of them. It's called mTOR, that if we turn mTOR down, we can actually modulate all of the hallmarks of aging and slow the biological aging process. So again, there's a lot we don't know. In fact, I sometimes suggest that our understanding of the biology of aging is comparable to what people understood about the surface of the Earth in 500 BC, right? So in other words, if you sail too far, you're going to fall off the edge. So we've got a lot to learn, but I also think it's useful to appreciate this map. This map was pretty helpful in 500 BC. It allowed people to explore. It allowed people to get rich trading, right? We understand enough, even if it's very early days, to actually, in a targeted way, modulate the biology of aging. And so that's what I'm going to spend the rest of my talk telling you about. Okay, so what do we know today? So hopefully you appreciate that aging biology is a real thing. There are biological mechanisms that drive biological aging. We know something about it. We know it's modifiable. We know we can turn it down or speed it up. We know that it's more effective than treating individual diseases in isolation, or what I would call reactive disease care, medicine 2.0. We understand some of it, the hallmarks of aging, useful but incomplete. We've got some biomarkers, so I'll talk briefly about biological aging clocks. Again, some of you may have heard of this. There are direct-to-consumer companies where you can go supposedly get your biological age measured. I say those are more for entertainment value, but they're really useful research tools. And I think they will be useful in a veterinary or medical setting in the very near future. So there are biomarkers where you can actually measure something about your biological age. And as I've already suggested, we know some of the nodes in this network, mTOR, and we know some potentially useful therapies to target that network. And I'm going to talk a bit about this drug rapamycin, which is a specific inhibitor of mTOR. So this is the drug that targets the protein or the gene. Okay. So rapamycin. Rapamycin is a super interesting molecule. It was actually first discovered on Easter Island from an expedition there that was looking at active compounds in soil samples. And so that's actually where the drug gets its name. Easter Island, also of course called Rapa Nui, that's where rapamycin comes from. It was found in soil samples there. And it's actually produced by a bacteria in the soil called Streptomyces hygroscopicus. If you look rapamycin up on Google, you might see it called a macrolide antibiotic, but it's not an antibiotic in the way we typically think of an antibiotic. So it has no activity against bacteria directly, but it will slow the growth. So it's anti-proliferative. It will slow the growth of eukaryotic cells, non-bacterial cells, and it's a pretty potent antifungal because it will slow the cell cycle. So rapamycin was tested for a whole bunch of stuff. It was researched as an anti-cancer therapy, as an antifungal. It ended up being first FDA approved by the FDA in 1999 for organ transplant rejection. So again, if you look in the clinical literature, almost all of the information you will find comes from organ transplant patients, where it has been used to prevent rejection in combination with strong immunosuppressants. You will also see it referred to as sirolimus. So this is one of those funny cases where in the clinical literature it's called sirolimus, and the research literature is called rapamycin, exactly the same molecule. And the brand name generic is called Rapamune. Okay, so it's used for several different types of organ transplants, first for kidney transplants, also approved for cardiac stents, some rare diseases, and there is a collection of drugs called rapalogs, which are structurally similar to rapamycin, have the same biochemical mechanism of action. that have been approved for other indications. And I may or may not talk any. If I say the word rapalog, you can think of it as the same as rapamycin. Okay. So we know a lot, though, because it's been FDA approved for 20 plus years now. We know a lot about its clinical use in humans in that context. People sort of first got interested in rapamycin in the longevity space in the sort of early 2000s, so 2003 to 2005. 4 different groups, including myself, sort of all independently hit on mTOR as a target that modulates the biology of aging in different organisms. And we know something about why that's the case. So people often ask, like, what does mTOR do? What does rapamycin do? And how does it affect aging? So mTOR is really one of the key sensors that helps every organism, at least every eukaryotic organism, make a fundamentally important decision, okay? So every organism from the beginning of time has to make this decision. Sense the environment and then decide, is the environment appropriate to reproduce, right? And so if you think about what in your environment is important to consider if you're considering having babies, right? So humans are complicated, but this is true for humans as well. But if you think of simpler organisms, there's only a few things that really drive that decision. Is the temperature right? Is the oxygen level right? And probably most importantly, is there enough food? Because if there's not enough food around, that's a really bad time to have babies, right? And mTOR is this sensor that senses the environment, primarily nutrients, and helps the cell decide, is it a good time to grow and reproduce? Or if there's not very much food around, mTOR shuts down, and that basically turns off the growth promoting signals and causes the cell to become stress resistant, okay? So that's what mTOR does, and it's an ancient evolutionary function. But it turns out that slow growth mode, low food, slow growth mode, high stress resistance also equals slower aging, because stress resistance is really important for combating the damage that goes along with aging. So we think that's why mTOR plays this conserved role across the animal kingdom of modulating the biological aging process. It wasn't selected by evolution to do that. It was selected by evolution to make sure organisms reproduce at the right time and grow at the right time. But it's this byproduct of stress resistance and slow growth that promotes slower biological aging. Okay, so we also know that in every animal where it's been tested, genetically turning down mTOR or treating them with rapamycin is sufficient to increase lifespan. That's true in single-celled organisms like budding yeast. It's true in nematode worms. It's true in fruit flies. It's true in mice. It's recently been shown in a non-human primate, the marmoset. All of those organisms, if you turn down mTOR, you increase lifespan. If you do it genetically, you increase lifespan. Or if you treat with rapamycin, you increase lifespan. It also seems to broadly improve health span. So it's not just increasing lifespan. It really seems to be delaying the diseases of aging. And I'm going to show you some real data to support that claim. Okay, so a couple of other things to appreciate about rapamycin that make it very interesting from the perspective of a drug you might think about giving to people or to pets to positively impact the biology of aging, to increase lifespan and improve health span. One of those is, it turns out you can start the treatment in middle age and still get most of the benefits. And this was first shown by the National Institute on Aging, that's part of the National Institutes of Health in the US, the NIA Interventions Testing Program, where they tested rapamycin in mice, 3 different sites, so there's built-in triplicate replication. And the first time they tested it, they started giving the drug to the mice at 600 days of age. That's about the mouse equivalent of a 20-year-old or 60-year-old person. Okay, so middle age for biological age for a mouse. And what they saw was that you get this robust increase in lifespan. So this is a typical survival curve for those of you who aren't used to looking at this kind of data, where the fraction of animals alive are plotted as a function of age. So when you see the curve move to the right, that tells you there was an increase in average lifespan or median lifespan right here. and an increase in maximum lifespan there of the population. So this is a collection of a few hundred mice in each group. So these are the controls that didn't get rapamycin. These are the mice that started getting rapamycin when they were biologically 60 years old. And you get this nice increase in both median and maximum lifespan. That's important because if you're thinking about something you might want to give to people, you don't want to have to give this drug to teenagers and count on them to take it for the rest of their life. We all know that's not going to happen, right? But it would be great if you could start giving it to people when they're 60, 70, 75, 80, if you could still get the benefits. So that's important from a translational perspective. It's worth mentioning, first of all, it's not a miracle drug. I'm going to tell you all sorts of good stuff about rapamycin. I don't want to imply it's a miracle drug, okay? It targets the biology of aging and has the effects we expect for a drug that targets the biology of aging. Full stop. But it is the most effective longevity drug we have to date. Do I think other things are out there that are better than rapamycin? Absolutely. I have no doubt of that. This is the best we've got at the moment for a drug that modulates the biology of aging. It's the most reproducible. It's been reproduced by dozens of labs at this point in multiple different animals, multiple different mouse strains. So it seems very robust. You can get up to about a 30% increase in average lifespan in mice. And as I mentioned, it appears to broadly delay aging phenotypes. And this is a collection of the tissues and organs and functions in mice where rapamycin has been shown to improve age-related outcomes. Now, what's particularly interesting with rapamycin is that in at least 4 different cases, you can see improvements in function in animals, aged animals, that have already shown an age-related decline in function. And that's true in the heart, the immune system, the female ovary, and the oral cavity, okay? You can take old animals where function is low or has declined and treat them with rapamycin for between 6 and 12 weeks and see that function rebound. And I'm going to actually show you data because I think it's pretty impressive. So I'll show you data for, I think I've got heart, immune, and oral cavity in here. Okay, so this is the data for heart. There have been 3 or 4 labs now that have all shown this. So again, independent replication by multiple labs. This is from Peter Rabinovich's lab at the University of Washington. He was the first to publish this. And so what they did here was they took young mice or old mice, they gave them echocardiograms, so imaging of the heart, and looked at a few different functional measures for left ventricular function. And I'm only going to show you 2. There's more in the paper. If anybody's really interested, the references here. So the 2 that I'm showing you here are left ventricular mass index and E to A ratio, which for those of you who aren't cardiologists, aren't familiar with echocardiograms, you can think of it as a measure of how well is the left ventricle of the heart contracting with each pump, with each beat. Okay, so young animals, old animals, and in each group, there's a before and after treatment period. So the treatment period here was 10 weeks. And in the old animals, they started the treatment at this 20 months of age. Again, about the mouse equivalent of a 60-year-old person. Okay, so let's look at the before data first. The before are these open bars here. So if you just look at young going to old, you can see there's a big increase in left ventricular mass index going from young to old. And there's a decline in function in E to A ratio going from young to old, okay? Importantly, before the treatment, there's no difference between the mice that are going to get the vehicle control, the placebo, and the mice that are destined to get rapamycin before the treatment. Those are the same. Okay, now what happens after that 10-week treatment period is really interesting. So again, the controls, the young animals didn't get rapamycin. The old animals that got the control, so the placebo, really showed no change in LVMI and showed a further decline in left ventricular function. They're getting older. That makes sense. But after 10 weeks of treatment with rapamycin, the old animals showed a decline in LVMI, back not statistically different from the young controls, and an improvement in left ventricular function back to not statistically significant, different from the young controls. So at least by these 2 measures of age-related heart function, 10 weeks of rapamycin was enough to restore function back to a youthful level, okay? So you can see an improvement in a short period of time in the heart. You can see an improvement in immune function. This is actually one of my favorite papers of all time. This is from Pen Zhang at the University of Michigan. And they did a really cool set of experiments here. So they were interested in understanding two things. How does aging affect the ability of the immune system to respond to a vaccine? This paper was from 2009, so way before COVID-19. We now all understand why aging of the immune system is really important, right, since the pandemic. They were interested in understanding how does the aged immune system differ in the way it responds to a vaccine, and does rapamycin impact that, okay? So the way this experiment was done was they took either young mice or old mice. They gave them a flu vaccine, an immunization against influenza, and then they waited 6 weeks. They either gave them a control or rapamycin, and then they challenged them with a lethal dose of influenza. So the mice that got the vaccine, if it worked, should be protected. If it didn't work, or if they didn't get the vaccine, it's a lethal challenge and they will die. And this is another survival experiment. Again, fraction of animals alive as a function of time after that lethal dose of influenza, okay? In this case, these are days. So the control here are the young animals that did not get immunized, young without immunization, okay? So when you don't immunize a young animal and you give it a lethal dose of influenza, guess what? It's lethal. All of the animals die within about a week, okay? This is just showing you the experiment worked in the sense that it was, in fact, a lethal dose of influenza. If you're a young animal and you get immunized, you're fully protected. Again, you can think of this as a control. This is just showing you that the vaccine worked, right? You got the vaccine, the immune system responded, complete protection against the virus. The first thing that surprised me is what happens if you're an old mouse. Again, this is about the mouse equivalent. In this case, they were 24 months, so a little bit older, maybe the mouse equivalent of a 65-year-old person, 70-year-old person. If you're an old mouse and you get immunized, that's this dotted line here. No rapamycin yet, just immunized. There's about a 2/3 chance your immune system is not going to respond to that vaccine sufficiently to confer protection against the viral challenge. I don't think it's quite this bad in people, but it's close. We know that old elderly people do not respond as well to a vaccine, and that's partly why when exposed to a virus, even if they've been vaccinated, they're more likely to get sick. Okay, so here's the striking other part of this, is if you're an old mouse that got immunized, but you also got rapamycin for 6 weeks, there's a dotted line up here under the solid line, complete protection. So again, by this measure of immune function, you're rejuvenating the ability of the immune system to respond to a vaccine, the aged immune system specifically. Only 6 weeks of treatment with rapamycin is enough to fully restore the ability of the aged mouse immune system to respond to a flu vaccine. So 2 places now where rapamycin seems as if it can restore function in an aged animal. And the third place, I'm not going to take you through all the data. This is work that was done by Jonathan Ahn when he was a graduate student in my lab that we published in 2020 in eLife, where we showed that in aged mice, we could also reverse periodontal disease. And we looked at 3 different clinically defining features of periodontal disease. Loss of bone around the teeth. So John actually did micro CT, whoops, I knew I would do that at least once. Actually used micro CT scanning to measure bone around the teeth. And he could see in the same animal, this animal was anesthetized, aged animals lose bone around the teeth. If the animal is treated with rapamycin for 8 weeks, you can actually see regrowth of bone around the teeth. You see a reduction in gingival inflammation, and you see a change in the oral microbiome that at least moves it partway back towards a youthful composition. We actually think that's mediated by the improvements in immune function, that corresponding change in the oral microbiome, although that's a hypothesis at this point. So again, we've got heart, we've got immune, we've got the oral cavity, 3 very different parts of the body. In all of those places, it seems as though, at least in mice, rapamycin can restore function that has been lost with aging. I'm not going to show you the ovarian data. I will just tell you there are now clinical trials for both periodontal disease, so John is running that in humans, and for premature ovarian failure in women, that's being run out of Columbia, asking whether rapamycin can have the same effect on these age-related declines in people. The last piece of data I want to show you is, and this is sort of leading up to my second talk, where I'll talk about the dog aging project. And part of what we're doing in the dog aging project is a clinical trial with rapamycin. One of the rationales for that clinical trial is this study here. This is a work that was done by Alessandro Bito when he was a postdoc in my lab. And the question here is very, very simple. So I already showed you that if you take 20-month-old mice and you treat them for the rest of their life, you can get lifespan extension. We wondered, could you get anything comparable to that by only treating them for a short period of time? Because we could see these functional rejuvenations after 6 weeks, 10 weeks, 12 weeks. We wondered if we would see effects on lifespan after only 12 weeks of treatment. And so Alessandro did this experiment where he took 20-month-old mice, again, treated them for either a control, placebo, or rapamycin for 12 weeks, and then stopped the treatment, put the mice back in their cages, back on the shelf, let them live out the rest of their life. Okay, we didn't do anything else to those mice. And this is the survival curve from the end of the treatment. So these mice are now 23 to 24 months of age at the end of that 12-week period. And again, what hopefully you can appreciate is the mice that saw rapamycin for 12 weeks in this particular group had a very large increase in remaining life expectancy. In fact, the increase in remaining life expectancy from just an absolute magnitude was longer than the length of time they saw the drug, which is kind of interesting when you start thinking about biological mechanisms, that you would get an increase in lifespan bigger than the length of time that the animals were actually receiving the drug. Now, obviously, we don't know if this is going to translate to dogs or cats or horses or people in the real world. And even if it does, we don't know if it's going to be a linear translation. But if we make the assumption that it will be a linear translation, that's about 2 to 3 years for a typical mid-sized 7-year-old dog, or about 2 decades for that 50-year-old woman I showed you at the beginning of my talk. So potentially pretty significant increases in life expectancy from transient treatment with rapamycin. Okay, so we're very interested, obviously, in knowing, does this translate to dogs and eventually to people? Yeah, but we don't know yet. What do we know? So what do we know about rapamycin in the real world? So, I mean, again, I told you about the organ transplant data. That I don't think we can look at to get any clues about biology of aging or longevity. The doses are completely different. The patient population is completely different. And those people are usually taking strong immunosuppressants. So I don't think we can be guided by what's known about rapamycin in organ transplant patients. What we do know is that in healthy elderly people, the rapamycin derivative, so this rapalog I alluded to earlier, called everolimus, seems to boost response to a vaccine similarly to what I showed you in mice from rapamycin. That's the work of Joan Manick, formerly at Novartis and a company called Restore Bio. Joan now has her own company called Tornado Therapeutics, all focused around mTOR inhibitors. And she showed in multiple clinical trials that in healthy older people, 6 weeks of treatment with a rapamycin derivative was sufficient to boost flu vaccine response. She also did a follow-up analysis. It's in this publication here, where she also showed that in people who'd gotten the mTOR inhibitors, they seemed to be protected also against coronavirus. They were not vaccinated against coronavirus because this was happening in 2019 before people were even really all that interested in coronavirus. So it seems as though rapamycin may have broader impacts than just boosting vaccine response and may more generally boost aged immune function, although that's a little bit speculative at this point. We published a paper last year for people who've been using rapamycin off-label. So what I mean by that is that in the United States, if a drug is FDA approved, a physician can prescribe that drug off-label, so not for the label indication, if they believe that it will help their patient. And so there are, I don't know what the exact number is, probably 10,000 or so people now taking rapamycin off-label because they think it may have health span or lifespan promoting benefits. And so we collected data from people using rapamycin off-label, and we compared it to people who'd never used rapamycin. So it's a very imperfect data set for sure. But we did find some evidence, first of all, that there was only one side effect that was greater in the people taking rapamycin. That was mouth sores, canker sores. That makes sense. That's the most common side effect seen in organ transplant patients. There were 5 other side effects that were different between the non-users and the users, but they were all better in the rapamycin users. So who knows? But one of the things that did jump out was that people who took rapamycin throughout a COVID-19 infection were less likely to have moderate or severe infections or to have symptoms consistent with long COVID. Again, matching up pretty well with the data that Joan had published. There is a paper recently published from a company called PetVet that rapamycin can reverse hypertrophic cardiomyopathy in cats. There's a lot of mouse data. I didn't talk about all the different diseases where rapamycin has been shown to be effective in mice, but both hypertrophic cardiomyopathy and dilated cardiomyopathy, rapamycin is effective in mice. So PetVet, I think, will get FDA conditional approval for this indication in cats for rapamycin probably next year. And there are multiple clinical trials, which I've already alluded to, happening right now. Periodontal disease, ovarian failure, muscle function, all in humans, and then the test of rapamycin in aging dogs, which I will talk about in my 1 o'clock talk. I do want to say, so I talked a lot about rapamycin. Okay, I studied rapamycin. I like talking about rapamycin. It's not the only thing in our toolkit. So it right now is the best use case, I believe, in terms of the magnitude of data and the degree to which it's been both reproduced and shown to have broad effects on aging. There are several other molecules, though, that have been shown convincingly, I believe, to increase lifespan in mice and in some cases slow the biological aging process, as evidenced by reduced disease, improved function. So this is a list of drugs that have come out of the interventions testing program. I mentioned to you, I talked about rapamycin. These other 3 in red here are particularly interesting because another study, completely independent, looked at prescriptions from the UK Biobank. So they looked at 100,000 plus people in the UK, looked at all of their prescriptions, and asked a very simple question. If we, for each drug where we have at least 500 people that have been prescribed the drug, and we compare them to roughly health-matched individuals, in the people who got the drug prescription, is there any difference in mortality, okay, in risk of death? And as you would expect, for most of the drugs, there was an increase in mortality, right? There's a reason people are taking these drugs, they're sick. But for these 3, which had come out of the mouse intervention testing program to increase lifespan in mice, in that study, mortality went down in the people who were taking these drugs, which is really interesting and unexpected. So does it prove these drugs increase lifespan in people, slow aging in people? No, it doesn't prove it. Is it a really intriguing correlation that points in the same direction? Yeah, it's really interesting. So I think there's reason to be optimistic with rapamycin and some of these other molecules, given that we know the biology of aging is so conserved that they are going to modulate aging biology the same way in people. In my mind, that's pretty much an open and shut case. I think the question is, when you get in the real world, which is a complicated environment, are they going to have any offsetting consequences that in the real world might not be apparent in the laboratory? And we don't know the answer to that yet. I think with rapamycin, we've got enough data from people using it off-label to be confident that it can be used safely, that the risks are very low. I know some people would disagree with me on that. I think it's just because they don't know the data very well. I think the risks with rapamycin are pretty low. We know the risks with things like estrogens are pretty low. We know the risks with, this is a class of drug called an SGLT2 inhibitor, canagliflozin. The risks are pretty low. So I don't think we're going to find serious adverse events from these drugs, but we don't really know until we do the randomized placebo-controlled clinical trials. Okay. So I'm going to just finish up the last 5 minutes or so and talk a little bit about Medicine 4.0, because this is what I'm doing now. So I have left my academic position. to found a company called OptiSpan, which is really about trying to create tools and technologies to bring Medicine 4.0 to as many people as possible. If you're interested, the website is here. There's not a ton of information on the website, but you can get a feel for what we're doing. So again, Medicine 4.0 is really about this. It's about pushing the health span curve out as far as we can, keeping people healthy as long as we can. So what differentiates, in my view, Medicine 4.0 from Medicine 3.0? And it's only a few things. One is geroscience, which I've just spent the last half an hour telling you about. So hopefully, even if you don't believe me, you appreciate why I believe this. I think geroscience is the future of medicine, and it needs to be the future of medicine. And I think artificial intelligence plays a big role here. And I know that sounds like everybody's excited about artificial intelligence for good reason. We've got a couple of specific use cases where we think artificial intelligence in the context of Medicine 4.0 can be a real game changer. Obviously, nobody knows exactly how this is going to play out. So one use case for artificial intelligence are these biological age clocks that I already alluded to. The idea that we can identify from complex data types signatures that are predictive of biological age or at least future health risks at the individual level. And so just to, again, give you a feel for how these things are created, the most common biological age clocks today utilize something called epigenetics, and they measure a specific mark on DNA called DNA methylation, okay? And it turns out that there are tens of thousands of methylated sites in all of our genomes. And those methylation sites influence how much a gene gets turned up or turned down, okay? So you can think of them as sort of controlling the expression of genes depending on whether that DNA at that gene is methylated or not. And if you look in enough people, you can find patterns within the DNA methylation, so a subset of marks that are strongly correlated with chronological age. You can predict a person's chronological age based on their DNA methylation pattern. But when you do that across the population, you have individuals who are off of this line. These people up here whose predicted methylation age is greater than their chronological age, and these people down here whose predicted methylation age is less than their chronological age. If it's the case, if you follow these people for 20 years and it turns out that these people get sick and die earlier and these people get sick and die later, that's potentially a predictor of biological age. And that's exactly what's been done. So people have used banked samples to show that using epigenetic age methylation patterns, you can predict with some degree of precision future health outcomes. And the people who are above the line seem to be older biologically. The people below the line are younger biologically. There are lots and lots of high-dimensional data types that we can look at to build these clocks. What we don't know at this point is exactly what they're telling us or what the best clocks are. And I think that's where artificial intelligence can play a big role here. One thing the veterinarians in the group might appreciate is that if you do the same thing with dogs, so you can use the epigenetic tests and look at dog age as well. When you do that and you look at biological age, it turns out dogs age faster epigenetically than people do, right? So this is human years here, dog years here. You can see a 6-year-old dog is like a 50-some-year-old human. But the super interesting thing, which I think matches up with all of our experiences, it's not linear, right? Dogs are aging biologically maybe 15 times faster than humans early in life, and then it kind of slows down late in life. The fact that you see this in the epigenetic age signature, and it matches exactly what we see in animals in the real world, tells us there's something real going on here. This is real biology that the epigenetic age clocks are measuring. In a collaboration with a computer scientist at UW, I worked with Sue and Lee. We used NHANES data. So NHANES is a human longitudinal study. So not epigenetics. We looked at physiological measures, blood parameters, even survey answers, socioeconomic status, put that all in a big model, and we were able to make a biological age clock based on that type of data. So you can do it based on all sorts of data types. People are even now starting to create these organ-specific clocks. So this is work from Morgan Levine when she was at Yale, where you can get different epigenetic age signatures for different tissues and organs. So I just want to give you a feel for where this is going. This is quite powerful if this holds up, because then in a single blood test, you can find out, maybe in me, it's my brain that's aging most rapidly, and in you, it's your kidney. Wouldn't that be great to know? So I think that's the direction that we're going with these clocks. Okay. Another use case for AI, and this is a company that we spun out of my lab called Aura Biomedical, is to find drugs that work better than anything we've got today. So this is where Medicine 4.0, its goal is. But I believe there are drugs out there that are much better than rapamycin, or combinations of drugs, or natural products, or gene therapies. We don't know about them yet, but if we can search for them at large scale, we can potentially push this survival curve out. So again, we can't do this today, but I think this is doable given everything I know about the biology of aging. I'm not going to tell you it's going to happen in 5 years. I'm not one of those guys who says that it's going to happen in 10 years. I don't know how long it's going to take. But I think the AI tools combined with robotics give us an opportunity to do massively high throughput longevity drug discovery, potentially find these things. And we're doing it as we speak. And then the last thing, and this is something we're beginning to implement at OptiSpan. I think in the not too distant future, all of us are going to have our own personal AI health span agents, where your individual biomarker data, health data, nutritional data, if you wear an Oura ring, your sleep data, your trackables from your Apple Watch, maybe even encounters that you have with your medical provider or with a coach, will all be the foundation for these generative AI tools to actually act as an agent for you to take control of your own health. Again, we're not there yet, but I think anybody who's been paying attention to these large language models and what's possible now can see this is not a pipe dream. This is something that I think will be here within the next 5 years. And I don't know if it'll be on our phones or if it'll be in the cloud, but I think that this is the direction where things are going, and there's a lot of opportunities. And I've been personally quite surprised, even in our very rudimentary uses of this within OptiSpan in our clinical program, some of the interesting insights that come out when you combine a coach and a doc and an AI agent with an individual. So I think there's a lot of reason for optimism. And I'll just finish up by saying I covered a lot. If you're interested in this topic, we have recently put out a podcast called the OptiSpan Podcast. This is the channel here. So I would encourage you to check it out. And I'll stop, and I'm happy. I only have a few minutes left, but I'm happy to take questions if anybody has any.
So my question is about in the beginning of your speech, you're talking about mTOR.
mTOR?
mTOR, yes. I didn't understand really about this. The really functional of it, because it's decision of growing the reproduction, yeah. or what? This I didn't understand very well.
Sure. So let me start at a high level. So mTOR at the cellular level, in individual cells,
yes.
is required for cell division, for the cell to go through the cell cycle and divide. And it directly senses amino acids and other nutrients. When nutrient levels are high, mTOR activity gets turned up. That promotes cell division.
Yes.
It also promotes organ size growth, and at the animal level promotes reproduction.
Till the moment that the organism needs to reproduce, to the moment that the maturity. Even in the adult body, every time a cell divides, you need mTOR. Okay.
And like muscle. So if you look in the fitness literature, muscle building, they talk a lot about mTOR because you need mTOR activation to build new muscle.
Oh, okay.
That's true in every cell, in the immune system, in the lining of your mouth, your gut. mTOR is really fundamentally central to promoting cell division. The reason why it's important here is when nutrients are low, it turns down those division promoting signals and instead induces the cell to become stress resistant, which is a slower aging state, we think.
Oh, so both is a good decision, both good decisions.
You need, it just has to be done at the right time, right?
Yes.
Just like cancer is an inappropriate growth signal, right? And mTOR hyperactivation is seen in lots of cancers.
So, okay. So the mTOR is an substance of the. It's a biomarker of the cells.
It is, although it's not a particularly useful biomarker because it does so much, right? In other words, there are better biomarkers that we can use to look at aging other than mTOR, because mTOR is so involved in multiple different cellular processes.
Yes.
But yes, it is a biomarker of growth.
To the moment, thank you.
Sure.